In this lecture, we will be focusing on the nephron, which is the functional unit of the kidney, and we'll specifically talk about how the nephrons work to filter blood and remove waste from our blood system, but we'll also talk about how they regulate water balance and ensure that we aren't losing too much water every time we create urine. So the nephron actually has three major functions in terms of removing waste from our blood, and we'll look at those three in a little more detail on the next slides. But those functions are glomerular filtration, tubular reabsorption, and tubular secretion. And I want to highlight that this figure from your textbook does a really great job at showing all three functions. And we're going to basically go through this figure in a little more detail to talk about those three functions: glomerular filtration, tubular reabsorption, and tubular secretion. So let's look first at glomerular filtration. So this specific process occurs at the renal corpuscle, and in the renal corpuscle we have arteries coming from our blood, and they create this kind of ball-like structure called the glomerulus, and surrounding that glomerulus is what is called the glomerular capsule. And this is where the first removal of waste begins at the nephron. Specifically, what is happening here is that blood pressure is really high inside these arteries and inside the glomerulus, and that high blood pressure is going to force small solutes from the blood into the glomerular capsule, and specifically that those solutes are going to end up in the space within the glomerular capsule. But in order to get to that space, there's actually three different layers that we have to travel through, that solutes have to travel through in order to enter that space. And that's what this figure here on the bottom is showing. So those three layers are first the actual endothelial cells of the capillaries. So solutes have to first pass out of the capillaries, out of the bloodstream. Then they have to pass through the basement membrane, which again is like a tissue that basically kind of connects these two layers of cells together. So that's our second step. And then finally, they actually have to pass through the epithelial cells that make up the glomerular capsule. So we have to have basically a lot of pressure that can really push these solutes, as shown by the green arrows, out of the glomerulus and into that space within the glomerular capsule. But once all of this material is here, we then refer to those solutes as filtrate. And that filtrate is what will eventually become urine. So just to kind of pull out a little bit, again, we've just talked about glomerular filtration, which again occurs at the glomerulus itself. At this point, what we've done is we have removed lots of different solutes from the blood. We've removed nitrogen-containing waste, we've removed glucose, amino acids, ions, and water. So a lot of stuff has been forced out of the blood. But there's actually some things in this filtrate that are actually beneficial, and we actually need those to be in the blood. And so that's where step 2 comes in, or tubular reabsorption. And so now we're going to move out of the glomerulus, and we're going to look now at the descending convoluted tubule. So in tubular reabsorption, which occurs in the proximal convoluted tubule, again, if we kind of follow this glomerulus, it's the first kind of descending loop that we see in the nephron. At this point, there again is material in our filtrate that we actually want to go back into the blood. And so things like water, essential ions, and nutrients, they're actually going to get pushed back into the bloodstream. Interestingly, about 99% of the filtrate that is pushed out of the bloodstream in the glomerulus is actually returned to the blood. So that means that only 1% of that filtrate actually is going to become urine. The proximal convoluted tubule is really nicely set up to kind of have maximum absorption. This region of the nephron, which I'm outlining in red, contains structures called microvilli. And those are the same structures that we've seen in the digestive system. Remember, basically what we have is that the tops of cells have these little tiny finger-like projections. And those projections allow for increased absorption. We've just dramatically increased the surface area of the cell, which means that there's way more space where nutrients can be reabsorbed. And table 16.1 shows basically how much of substances are filtered and how much of that actually gets returned to the blood. So notice things like water and glucose, almost 100% of those things are actually reabsorbed back into the blood. Same with sodium ions, potassium ions, and bicarbonate. So we push a lot of stuff out in the glomerulus, but we reabsorb that back in. Things that we do want to keep in the filtrate include molecules like urea and creatinine. Those things don't get reabsorbed as much. So at this point, we have pushed everything out in the glomerulus. We've now reabsorbed some material that we want to keep in the blood. Referring back to this diagram, the last step that we want to perform is tubular secretion. And so this is where we're basically going to further concentrate that filtrate. We're going to further make sure that what we don't want in the blood is leaving. And let's talk about kind of what is happening during this tubular secretion process. So the last stage of filtration by the nephron occurs at basically all throughout the renal tubule, but primarily we'll see a lot of this happening in the distal convoluted tubule, but this can occur both at the proximal convoluted tubule and at the collecting duct as well. So it can really happen kind of throughout the nephron. What we are doing during tubular secretion is we are working to remove additional waste and excess ions. So ions like protons, H+, potassium, and NH4+, those things are going to be secreted into the renal tubule because those are things we want to get rid of. This also is where foreign substances like pesticides and drugs can also be removed from the blood so that we can further purify the blood. But together, glomerular filtration, tubular reabsorption, and tubular secretion all work to do one thing. And that is that by the end of filtration, blood has been purified. It can then go back and return to the body, pick up more waste, and come back to the kidneys later. And at this point now, the filtrate, that is the fluid or kind of concentrated substance that is now in the tubules, that is now considered urine. And urine is going to travel down the collecting duct as shown by the black arrows. From there, it'll move to the ureters and then to the urinary bladder, where this will be stored until the urination process. So the biggest kind of function that the nephron does for us is to filter out waste from our bloodstream so that we can get rid of those dangerous particles and ensure that our blood stays nice and clean and has all of the materials that it needs and nothing that it doesn't need. But one thing we haven't talked about is what happens at the loop of the nephron. And specifically, one thing we haven't talked about that is a really critical function of the nephrons and therefore the kidneys is water conservation. So about 20% of our nephrons have these really long loops that dip into the renal medulla. So they dip into kind of the deepest parts of the renal medulla. And these loops function to reabsorb water. And when water is reabsorbed, this helps to concentrate urine. So you've probably noticed that if you drink a lot of water and then go pee, you have pretty clear urine. But if you drink water and then you don't pee for a long time, or maybe you didn't drink a lot of water throughout the day, your urine is much darker. The darker your urine, the more concentrated it is. By maintaining and reabsorbing water, by maintaining water balance, this also helps the kidneys to work with the circulatory system to maintain cardiac output and to maintain blood pressure. So it's not all about just conserving water. It actually does help other systems as well. And again, really all of the concentration or reabsorption of water is going to occur in this region, in this loop region of the nephron. So let's actually look at what's happening there. So first off, again, water is traveling in that filtrate from the glomerulus, and once it enters the loop of the nephron, it's going to start to actually leave the filtrate by osmosis. So these blue arrows here that I'm circling represent water leaving the nephron and entering the interstitial fluid that surrounds this region. Remember that if water is leaving by osmosis, remember that there is an inverse relationship between solutes and their concentration and water concentration. So remember, for example, if you have high concentration of solutes, you have low water, and vice versa. If you have low concentration of solutes, you have high water. So if water is leaving the nephron, that means water in the nephron, water concentration in the nephron is high compared to the environment, compared to the interstitial fluid, it's low. That means that in the nephron, solute concentration must be low compared to solute concentration outside. So we have, again, our inverse relationship where in the nephron, we have high water, low solutes, and in the interstitial fluid, we have low water, high solutes. This is important to keep in mind as we talk about the water reabsorption process. So in the descending portion of the loop, this area is permeable to water, and so water will leave by osmosis. And eventually, because now that water is in the interstitial fluid, it can be taken back up by the bloodstream and returned to the blood. From here, we're going to move into the ascending loop. And now these blue arrows don't represent the fact that water is leaving, they instead represent the fact that salt is leaving. So these blue arrows, let me pick a bit better color, actually represent the leaving of NaCl here. So what's happening here is the ascending loop. A big key thing to keep in mind is that the ascending loop of the nephron is actually impermeable to water. Water cannot leave through this side of the nephron, but what can leave is salt. And specifically, NaCl is going to be actively transported. So it's actually being pumped out of the nephron and into the interstitial fluid. It's actively transported, there we go. At the same time, this is also allowing urea, which is shown by the green arrows, to enter the nephron, and that's good. We want urea to be part of urine, so it allows another molecule to move in and become part of the filtrate. So descending loop, water has left. Ascending loop, water cannot leave. It is impermeable. The ascending loop is impermeable to water, but we are actively pumping out sodium. And that's important because that active transport, it's going to keep solute concentration high in the interstitial fluid. And that is really important for the last part of water reabsorption. So finally, if we continue to follow the black arrows throughout the nephron, we have a final place where water can also again be reabsorbed into the interstitial fluid, and that is at the collecting duct. So these blue arrows here again represent that water is leaving from the collecting duct, and the collecting duct can change its permeability to water. So sometimes it can be more permeable than others, and that is because of the hormone ADH, or anti-diuretic hormone, which we've talked a little bit about before. However, if you have anti-diuretic hormone that in your bloodstream, that's going to promote water leaving. So if you have more ADH, you have more water conservation. So that's going to promote water leaving the nephron, which can further concentrate urine. Again, water, if the collecting duct is permeable, water is just following the laws of osmosis, where it's moving from high water concentration inside the nephron to low water concentration in that interstitial fluid. And that's where I want to take a second to go back to step 2. Again, remember the whole point at step 2 is to really pump NaCl out of the nephron and into the interstitial space. And again, by doing that, that keeps the solute concentration high outside of the nephron, which ensures that we're always promoting the fact that water wants to leave the nephron so that we can concentrate urine. So we're always taking advantage of the process of osmosis to regulate the concentration of our urine and the conservation of water. I also want to point out that the longer that our nephron loops are, the more water that can be conserved. So if we have pretty shallow loops, our bodies won't be able to conserve that much water. The longer the loop, the more time basically we can allow for water to leave the nephron, which can further concentrate our urine. And just kind of a fun fact about this, many animals that live in the desert, for example, have really, really ridiculously long nephron loops, and that allows them to conserve as much water as possible when they live in a really hot and very water poor environment. Animals that live in kind of more lush areas that gets more rainfall are going to have shorter nephron loops. So just kind of a fun fact about nephron loops. But again, we've now seen how the nephron is the functional unit of the kidney. We've seen how through 3 steps it can regulate the removal of waste and filter our blood to make sure that our blood stays waste-free. And we also see how it regulates water conservation through using the specific properties of osmosis in the nephron loop. Thanks for watching this, and reach out with any questions.